First of all, before we start, I'm George Carlson. I'm with the McLaughlin Research Institute. And I'd like to thank the Benefits Health System for letting us use this auditorium and for setting this up on the REACH network so people can see it at the Poseidon Cancer Institute and all across the state. And it'll be available for viewing at medical sites throughout Montana after this is over. Now, this talk is sponsored by the Howard Hughes Medical Institute, who gave a grant for K-12 education to McLaughlin Research Institute. The Howard Hughes is the largest private funder of medical research in the country. And they are very interested in promoting pre-college education as well as college education in the sciences. So as you know, as many of you know, we have high school students and high school teachers coming to the laboratory each summer that are funded by this Howard Hughes grant. And it also funds these public lectures. Now, for example, I think many of you may have come to our talks on evolution and on systems biology last year. And you might ask why, if we're interested in K-12 education, we sponsor high school students and teachers, why would we do a public lecture? Well, there's several reasons. The first, science and technology become increasingly important in our societies, forming a larger and larger sector of what's important for growth, economic growth, for knowledge, for medical care, and we have a smaller and smaller proportion each year of citizens who are expert in science and technology. So it's a collision course. We're going to be more dependent on this, and if we have fewer people who are able, the United States won't be as competitive with other countries that are investing in research and education. So we're trying to increase scientific literacy. And the second part of that, we want to increase your support, the support of the public, for science education programs in our schools, which is very important to get people to choose careers in science and technology. The third, and I think in my opinion the most important reason, is we really would like to foster critical thinking among the general public. And it's not important just for science, but to be able to evaluate data, or translate just that as information, any information, as for the accuracy of the information, for the source of information, to be able to guide you in evaluating, not just accepting what you're told, but thinking about what you're told is a normal part of life. Now, our speaker, Irving Weissman, for many, if not most of you, needs no introduction, even for those who didn't happen to go to school with him. He was born and raised in Great Falls, and again, most of you know he got his start at the predecessor of the McLaughlin Research Institute doing research under Dr. Ernst Eichwald. And he's been grateful and supported ever since. He also volunteers as chair of the McLaughlin Research Institute Scientific Advisory Committee to help us evaluate the research project and set research directions to make sure we're current with the cutting edge. And some of you may think of him as the person who brings the ski and ballet to Great Falls every other year. So it's through his generosity to the ballet, funding that, bringing that in for the ticket sales of any benefit involved. So he's more than giving back to the Institute and to Great Falls. But some of you may not, you sort of vaguely know what Irv has done in his other life. But after graduating from Montana State, he went on to Stanford, and I guess they wouldn't let him leave. And he's been there ever since. And his awards and mention are just, and honorary degrees and so forth, too numerous to mention. He was elected as a member of the National Academy of Sciences in 1989. He's gotten honorary degrees from a variety of places. He led the National Academy's panel on stem cells and the ethics of stem cell research. He founded at least 2 companies based on the stem cell research, Systemics and Stem Cells, Inc. And all this is in recognition of what he did in identifying and isolating, not push with bone marrow cells for transplantation to actually isolating the stem cell. And he's going to be telling you some of that today. So that was the first person ever to isolate a blood-forming stem cell. He wasn't content with that, and after Brooks went on and isolated the brain-forming stem cell. He's currently director of the Stanford Institute for Stem Cell Biology and Regenerative Medicine. He's the Ludwig Professor of Cancer and Clinical Research, as well as a professor of pathology at Stanford University. And I'd like to welcome Irving and also to thank him for all he's done for Great Falls.
Thanks, George. Are we going to have a slideshow here? Good. Is it up there yet?
Yep.
Good. All right. So first I want to set the record straight. As many people know, never once in my career in Great Falls schools did I make the top 10% of my class for grades. Jack likes to point that out because Jack was way above me. But I was lucky enough when I was 16 to meet with Ernst Eichwald at the Montana Deaconess Hospital and start a job in the lab so I could find out how the information that led to what was in textbook was obtained. I was never good at memorizing information, but I found out that I was okay at doing research to figure out how things work. So I'm going to talk to you a lot about how things work in the body, and it'll seem a little scientific. I hope it's not too scientific. The only person I'll ask real tough questions of at the end is Jack Weber, who I'll pick on. And of course, if he falls asleep, he's dead. I know. That's why I said it. So the only word that I'm worried you don't know is the word cell. So cells are the organizational units of the body. Every organ, every tissue are made up of cells. And in our bodies, which have to last a long time, we have a special kind of a cell that is entirely responsible not only for the first development of the tissues and organs, but their maintenance throughout life. Our bodies are being regenerated all the time from stem cells. Now, the definition of a stem cell is actually very simple, and it's amazing that people missed it for so long. A stem cell is a cell that when it divides to give rise to two cells, at least one of the two is still a stem cell. So if it was a skin-forming stem cell, one of the two would be a skin-forming stem cell, and then the others would go on and on to make parts of the skin, parts of the hair follicle, parts of the glands that moisten the hair follicle, and so on. But the cell that's at the base of it is the skin stem cell. And the task we had was to find that very, very rare cell. When we finally found it, in the blood-forming system, it was 1 in 15,000 cells in a blood-forming organ. Like the bone marrow is where all of us are making blood. Earlier in life, we made blood in a place called the spleen. Even earlier than that, in the liver, during fetal life. So the stem cell that makes up and forms the blood-forming system was born during fetal development and has sustained itself. There's no new stem cells coming in, so you're living on the stem cells that you had. We first tried to isolate the blood-forming stem cell for what now turns out to be obvious reasons. First, those people who died in Hiroshima and Nagasaki after the bomb, not by the blast, not by the fire, but by the loss of a tissue function, died at the lowest dose of a radiation because they couldn't make blood. And so they couldn't make this kind of cell that makes platelets that clot the blood. They couldn't make these kinds of cells that fight off the infections that you carry with you but are normally not disease-causing. But as soon as you lose those cells, then you've had it. In the practice of medicine, we need to regenerate the blood-forming system quite often. Over the last 20 years, one of the reasons to do bone marrow or blood-forming stem cell transplants is for somebody who's got cancer and the cancer spread beyond what a surgeon or radiation therapist could take out. So it's gone, it's spread, it's called a metastasis when it spreads, and it takes residence somewhere where you don't even know where it is. So it means you can't cure the cancer with surgery, you can't cure the cancer with radiation because you can't do the whole body for the reason I told you. So they give chemotherapy, and they usually give combinations of drugs because even before you give a drug, already 1 in a million of the cells is resistant to first drug, and another 1 in a million to the second, and another 1 in a million to the third. But no single cell is already resistant to all three. And I won't go into why that's so, it's just so. So you have a chance of curing a cancer if you can get 3 or more drugs that act on the cancer cell, but every one of them has side effects, and by the time you get to the dose that'll cure the cancer, or has a chance of curing the cancer, you've wiped out the blood-forming system. So the practice of bone marrow transplants from yourself to yourself developed. And when it was found by us and others that, for example, a woman with breast cancer that is spread beyond the breast, so it might be in the bone, it might be in the liver, it might be in the lung, and you know you're going to have to do whole body chemotherapy, by the time that's happened, the dose of radiation you would give would kill off the bone marrow and the blood formation. So people would take the bone marrow or another thing called mobilized blood. It's simply a way to mobilize the stem cells out of the bone marrow where they usually like to sit and into the blood. And instead of having somebody take you to the operating room, stick a needle in your hip 150 times to just barely get enough stem cells to regenerate your system, you can stick a needle in the arm for 4 hours and get more than enough. Because all the stem cells in the body, for a while, get mobilized in the blood. Then they know how to go back. And it's that knowing how to go back that's important. But you might imagine you take all of these cells, including cancer cells, you treat the patient, and you give it all back. Those that had cancer cells that contaminated the bone marrow or the mobilized blood are going to get the cancer spread again. And this is just a small comment on education. When I went back to say to the oncologists who treat breast cancer that we wanted to have a way of giving back cancer-free stem cells to regenerate the blood, they say, how do you know that if you put cancer cells back in, they'll seed the body and cause cancer? These were oncologists who were the best of the people, who did get straight A's and could memorize like mad, but didn't always understand how information was obtained. And this is all about how you get at understanding what's going on. And so I would answer them, well, you wouldn't know unless you had studied a century of tumor biology, where we all know that's exactly how you understand what a cancer is. You put it in the body, and if you put it in the bloodstream, it'll land somewhere, and it'll grow a secondary or metastasis, a spread. So anyway, I had to form a company in order to identify and isolate purified stem cells, and that's worth discussion at some point. But I'll just say that the company is not there anymore, so I'm not biased in what I say. I'm not trying to preach for you to go get this treatment or not. So we developed a treatment. We had 22 women at Stanford and at Detroit who had cancer that had spread. We isolated their blood-forming stem cells, and every patient now had blood-forming stem cells free of the cancer because we had the pure population of stem cells. They then got very high dose chemotherapy, and then they got the stem cells back. And this is actual data of cancer-free survival. So they were alive and they had no evidence that the disease had come back. And this was overall survival, so that's a pretty high fraction, narrowing down as the cancer got to them. This was a group on the East Coast that gave back everything, including in 40% of the patients, cancer cells. Now, if you'd seen this, you'd say, that's what you should do. But one of the lessons that I learned, the hardest lesson, was right at this time, a company bought us, a very big company. And they decided, before seeing the data come out, that there was no chance on earth they could make their minimum requirement, $500 million a year in 3 years. So they shut it down. So this is what the standard of care was and probably is. And you could ask me later what else was going on. I'll just say, sadly, about 4 years ago, one of the executives who made that decision that it wasn't in their top priority called me and asked me about cancer therapy because his wife had breast cancer that had spread. So, from the discovery of the stem cell, mice in 1988, humans 1991, clinical trial starting 1996 was a pretty short time. About the shortest time I've seen where you go from discovery to a therapy. But there's always stuff in between that you don't expect, you don't understand till you get there. And overcoming that is difficult. We're getting back the ability to do that at Stanford next year because the patent will run out. And the company will have no reason to hang on to it anymore. I'll add a couple more like this. I don't want to disappoint you completely. So, but there are conditions where you want to be able to take the blood-forming stem cells from somebody else. Now, if you imagine that, since I'm using them a lot, Jack Weber is my brother, and I promise you he's not. And let's say he needs to have a transplant because he took some wrong drug and lost his blood-forming system by the toxicity. And I was going to transplant my blood-forming tissue into him. The problem is that I already developed immune systems that would reject a skin graft from him onto me, or a heart graft, or a lung graft, or an insulin-producing islet graft. So I have immune cells, they're called T cells, that are responsible for rejection of his tissues. And now if I put my whole bone marrow in his body, those T cells that would fight off the skin go to his skin and make sores. Those that fight off the lung will make huge inflammatory lesions, so he can hardly breathe, and so on. So the trick was, how can you transplant blood-forming stem cells, tissues, without getting that disease where the donor reacts against the host? And the answer was, it turned out, pure stem cells. They don't have any T cells. So now this is me, this is Jack Weber. We irradiate him. Then we transplant him with my pure blood-forming stem cells. Now he has my blood-forming and immune system because it came from me. And my stem cells know how to make more stem cells. So if the transplant takes, it's one time for life. If we do it the right way, there's no drugs needed to keep it in. Here's an interesting thing. My blood-forming and immune system in his body still recognizes me as self. So you could do a skin graft, or a heart graft, or a lung graft from me to him the same day or a year later, and it would take. because it's my blood-forming system that cannot make an immune reaction against me. And when they develop the new T cells that are fighting off infection, they won't make an immune response against him. As a consequence, and this is just the mouse experiment we did about 20 years ago, you can co-transplant a heart and a blood-forming stem cell into this mouse, and it takes for life without any immunosuppression. Now, my blood-forming system in his body still recognizes a third person's heart, as foreign. And we've shown this not only, in mice, but mice that have human immune systems that we restore with somebody else's stem cell, and now, in a preliminary way, in the clinic. Now, let's say Jack had juvenile diabetes. That's not a disease of the insulin-producing cells. That's a disease of the blood-forming cells. There's a mistake in the blood-forming system so that it now makes an immune response against the insulin-producing islet cells in the pancreas. It kills them. Now, we don't know the whole genetics of it, except that we know it's inherited. But we know this now, that you can transplant blood-forming stem cells from a diabetes-resistant donor to a host that's about to get diabetes, first in mice, and we'll move it to humans. If you give them their own stem cells, they still will cause diabetes because you got the same mistaken blood system. But here we published in I'd say probably 96, 97, that a single transplant of stem cells into a mouse that was just starting to have juvenile diabetes prevented the disease for life, because you not only had the resistant cells in, but because they self-renew, they last for life. But what about the diabetics out there? They already lost their insulin-producing cells. So we co-transplanted, Judy Shizuru and I, blood-forming stem cells and insulin-producing islets. And we could cure a mouse at any stage. So we're at the stage that we're ready to take advantage of stem cell biology and move it to therapies. We just need to get past some of these barriers. We've shown these autoimmune diseases, which are in this audience, I assure you, are also the same thing. Genetic-based autoimmune diseases, and there are resistant stem cells, blood-forming stem cells, that make blood and only blood, that can, in the models in animals, stop the immune part, the autoimmune part of the disease. Lupus, rheumatoid arthritis, multiple sclerosis, and so on. So we need to be able to move this as fast as we can to the clinic. And we're dealing with a lot of barriers. No pharmaceutical company understands or will, in this days, finance a stem cell company. Because for them, self-renewal means that you remember every day to open the drug pouch and take your pellet and go on the next day where you're going to need it again for your chronic disease. No biotech company that did proteins, like Amgen for erythropoietin and so on, has the mindset to do it also. So every step of the way you have to not only be involved in the invention, and then what we call the preclinical phase, showing a proof of principle that it should work for the disease you're going to do, but then you got to cover that barrier, because in America and everywhere else, drugs and therapies are commercialized not by universities, but by companies, of course. So you have to form a new kind of company. We thought we were there with systemics, but we got bought out. I said that to my brother-in-law, who probably still has the stock. Sorry, Gene. Anyway, so I'm going to show this part. I wasn't going to show this, but I think there's a neat connection here. So it's an interesting thing that as scientists get older, they start to study aging. You know, just maybe it was luck, maybe it just turned up that way. I don't think so. And so we had stem cells. And we wanted to know, well, what happens to an aging blood-forming system? So we could isolate blood-forming stem cells from a young mouse or a young human, and blood-forming stem cells from an old one. You can already see something is different. The blood-forming stem cells from an old person, instead of having fewer of them, which we think, you know, before we did experiments, everybody said, well, aging is a loss of stem cells, as if they knew anything about it, because they never had a stem cell, so they couldn't do an experiment. But it was taught, aging is a loss of stem cells. But no, you have 2 to 5 times the number of blood-forming stem cells as a young mouse or person. And that's like the Red Queen said to Alice. Sometimes you have to run twice as fast just to stay in the same place. Well, that's us, aging folks. So, we found, oh yes, somebody's thing is going, and I'm going to shut mine off because you don't want to listen to Willie Nelson. Okay. We found out that old stem cells behave as old stem cells in a young or old body. So they still make more cells than putting young stem cells into the same mouse. So they've changed what they do. And somehow they all have to know what change they have. And at a more sophisticated level, old stem cells in mice and humans tend to make more blood that can fight off an infection by immediate recognition, cells called macrophages and granulocytes. Whereas the young, you tend to make those cells as well as lymphocytes that when you're vaccinated become your memory. So old people and old mice have already met most of the infections they're going to meet. They're living on memory, and some of you can imagine what else you think about when you're living on memory. Few of you might remember that. Young folks don't have any idea. So, Isabel Bierman, a McLaughlin Research Institute student about 10 years ago, now doing her postdoctoral work after her PhD at Harvard, joined up with our group to do an experiment about aging. And we had a very simple point we wanted to get at. When you have young blood-forming stem cells that are going to turn old, does every cell learn how to age and change what it's going to do? That would be pretty amazing, knowing what we know about how cells work. Or do you have a diversity of cells from birth? And the ones that are suitable for aging are not so competitive in the young person, but they get super competitive for bone marrow sites when you're old. And the result of Isabel's experiment was there's a tiny number of stem cells in the young that become the dominant number in the old. And for those biologists in the audience, if all the stem cells had changed their genetic expression to become old, that would have been, in a sense, inheritance of acquired characteristics, like Lamarck. But if you have competitive stem cells, some of which can survive natural selection better in the young, and others of which, when you're old, that's Darwinism. And as happens almost every time when you can make the question come out, Lamarck versus Darwin, Darwin wins out. Won this time for sure. Okay. So, after the egg is fertilized, it looks like that. Now, in humans, that happens is the egg pops out of the ovary and starts going down a tube called the fallopian tube. The sperm race up and fertilize it. It then starts to divide. And it goes from 1 to 2 to 4 to 8 to 16 to 32, 64, 128. And about 128 or 256 cells it becomes a ball of cells like this. It has not yet attached to the uterus. It cannot go any further until it attaches to the uterus. But when it attaches to the uterus, these cells on the outside are going to form the placenta, and these cells on the inside, each cell could participate in forming all of the tissue and organ systems. So they're called pluripotent, many potencies. And they last not very long, because at the time that you really start forming the layers in the tissues, then you form the stem cells for all the different kinds of tissues that are going to maintain you for life. Now I'm going to come back to that concept. You go from pluripotent to fewer potencies, or multipotencies. So one of the popular experiments that was published by all the lead journals said that blood-forming stem cells are only blood-forming if you get them out of the bone marrow, the blood-forming organ, and that they'll naturally move to the liver where they become liver stem cells, or to the heart where they become heart stem cells, or to the brain where they become brain stem cells, or to the muscle when they form muscle stem cells. And since the time they were saying it, I still had a company that had the only commercial right to transfer blood-forming stem cells. I thought, boy, I hope it's true. And so we tested it. We found that blood-forming stem cells make blood, but they don't make heart or skeletal muscle or lung or brain. Brain-forming stem cells, which we later found, make brain and nothing else. What I have in this little diagram is about 6 years of work by Amy Wagers and a team of scientists. So that set up a different principle. Once a stem cell is specified for a tissue, it can make that tissue and only that tissue. So this is a list of stuff that you don't need to read. It's just to say, how do you know when something's a fact? And it has to go through this whole set of things for proofs. But they're pretty standard, they're pretty stringent, and if they weren't true, we'd be heating our houses with cold fusion. Everybody can make that first guess, that first idea, and maybe even get it published. But when independent people try to verify it, who have no stake in the game, often you find out it's just not true. And the mistake could be not of intent, but just a mistake. Or it could, unfortunately, sometimes be by intent. So when it turned out blood-forming cells couldn't turn into muscle, an undergraduate in my lab and I, Richard Sherwood, isolated the muscle-forming stem cell, and when we put it through its paces here, so that's the muscle-forming stem cell, here's a couple of other cells in the muscle, the only one that could regenerate the muscle in muscular dystrophy was the stem cell. The next step down can't do it. So only the self-renewing cell can regenerate the whole tissue. Now, there's lots of good and bad about that. We've got the cell. We know that the mouse muscle stem cell cures the muscular dystrophy that has exactly the same genetic defect as kids with muscular dystrophy. That's the good news. The bad news is if you put a muscle cell in the blood, it doesn't make it to the muscle. That's why it was so lucky that blood-forming stem cells from the bone marrow put into the blood knew how to home back to the blood-forming tissue. So a muscle-forming stem cell could regenerate the muscle for life of somebody, but you'd have to find a surgeon who'd want to do all those transplants to all the major muscle groups in the diaphragm. Well, luckily, if you know surgeons, especially in academia, that's exactly what they want to do. So we also isolated the human brain-forming stem cell, and this was also at a company that we co-founded. And again, it was because we knew we wanted to move as rapidly to translate this to medical treatments, and at the time, that wasn't done at a university. Now, things are going to change, as I'll show you in a couple of minutes, but at least at the time. So we found a single cell that could make, in a test tube, thousands of cells in the brain lineage. And we tested whether they were brain-forming stem cells, not by getting a bunch of medical students and putting them in their brains, but into mouse that have no immune system. And we found that the brain-forming stem cell put here spread throughout the brain of the mouse. And if you had put in a mouse brain-forming stem cell, it does exactly the same as the human brain-forming stem cells. So they multiply for life where stem cells find their home. They migrate long distances where you need to migrate, from the source of stem cells to where you need the new neurons to affect smell. And in mice, it's a big area. In us, it's a little tiny area. You have to have the balance and thinking, and you need to transmit signals rapidly down a nerve, and you can't do it unless it has insulators spaced down so the electrical signal can jump rather than move slowly. Now that's a long way away, but I think you'll get this part. I've lost part of it, but don't worry. There's a childhood disease called Batten disease. Those children are perfectly normal, except they lack in one gene of all the genes in the body, one amino acid as part of the protein. That's because they have a mutation in a gene. That gene makes a product that cuts fats that are connected to lipids. Now, fatty lipids are lipoproteins. Fatty proteins are lipoproteins. Wear out, and like everything else in the body, have to be reutilized. So there's a garbage can inside of every cell where you break down the stuff, and then you break it completely down, and then you use the building blocks to rebuild proteins, and so on. Well, they can't start the process because they lack the enzyme that cuts the fat from the protein. That's it. They're normal until they're 2 or 3 years old. Then they start having visual symptoms. They start going blind. Then they start having balance problems because the cerebellum has the balance center. The cells are starting to die. Then they start losing intelligence as the place that you need to have new neurons born to make short-term memory. So I'm going to say that again. There's a place in the brain in everybody in this audience that makes new neurons over 30,000 a day. And they're responsible for me saying this to you, you hearing it and putting it into short-term memory. If I have a lesion in that part of the brain that kills it, and I turn away and look here, I can remember everything about you 50 years ago, but you can't remember what you just learned because those cells are responsible for short-term learning. So they start losing that next. And they go into a coma next. And then they all die. This is the brain of a child, 7 years old, with that disease. This is what the normal brain at that age would be. And you see these great big holes because the brain substance is gone. Now we found out that that was not only a disease of one enzyme, but it was an enzyme that one cell could make and send over to another to clean up the garbage dump. So that's a neat system. So we looked at a mouse. You'll start to get this theme over and over again. It's the theme of the McLaughlin Research Institute. Take the gene that causes the disease in the human, look at the mouse equivalent, make the same mutation. Now you got a mouse that this is the place where you learn. So this is for the experts only, the hippocampus. I don't understand why it was called the hippocampus. It should be said the learning center, right? So here's a normal hippocampus with these two parts. This is where the brain stem cells are born, and they project up to here and here and here to keep these cells going and make new learning. This is the Batten's disease mouse. And you can already see huge numbers of cells are gone. So we took human brain stem cells and put them into a Batten disease mouse that had no immune system because we had bred the mouse so it wouldn't have an immune system. And the more cells you put in, the more of those cells you protect by making the enzyme and sending it to those other cells. And we've transplanted 6 patients at the University of Oregon. And we did it at the University of Oregon not only because it's a great place, but another famous institution I know well had a bioethicist or a lawyer say, well, you can't do the first human brain cell transplant in a child, so you can't do it here. And if I'd been in the room, I'd say, yeah, but nobody gets out of this alive past childhood. So you're saying that you're not going to take care of people with that disease because you're afraid of being sued, or you think it might be wrong? So again, you come up against barriers. Now we knew you had to go to more than one hospital site to do a trial if you were intent on trying to treat this disease. First 6 patients passed. They had to give us patients that were in late stage. Nobody suffered from the transplant, so now the FDA and the institution are going to let them do early onset patients where we think we can do something about their lives. Spinal cord injury. So, in the contrast to the samurai novels, where you get sliced through the spinal cord, when you have a spinal cord injury like Christopher Reeves, you have, this is a skeleton not to freak you out, but just to show you where. You have the 9th thoracic vertebrae. It's very susceptible to a crush injury because the impact. And at the area of the crust, the cells that had their cell bodies there die. And you lose motor functions in the hind limbs because you may think about moving your leg here. Now, in all of us, that happens immediately. But it can't get past this area because the transmission is gone. And if you have pain in the lower leg, the transmission to the brain to say, it hurts in my left lower leg, is gone. So we transplanted human brain stem cells into immune deficient mice who had the same spinal cord injury. And we put them just above and below the injury. And in blue, this is a score. It's blue on mine. It's purple or whatever. You see the behavior of the mice over time. And this means they're walking in a coordinated fashion, front and hind limbs. And this got not brain stem cells, but another kind of cell that some people are selling on the street, called mesenchymal cells, does nothing. Walking, paralyzed. After 16 weeks, 9 weeks of walking, we put in a poison that kills human, but not mouse cells. Paralyzed again. And if you look, what happened was those insulating cells called oligodendrocytes that allow the electrical circuit to go fast had been killed because they were in the region that lacked blood during the crush, and they were replaced with human cells that allowed walking. And we're scheduled for a clinical trial, probably in Europe. Now, I'll just say this, and I know everybody's going to piss on me, excuse me. A country that has National Health Service, so that you don't negotiate with each insurance company separately to try to do a clinical trial. Now, whatever your choices are, just remember this. If we're going to do clinical trials, we have to get a large enough group to prove or not prove the principle. And you can do it in a country that has National Health Service, and it's very hard to do in America. We are way behind in the capacity to do clinical trials, except for people who are dying of a disease, and it might save the insurance companies if you could cure them. So now, we're going along fine, and I get a notice that Senator Sam Brownback has just written a bill saying that if you put human brain cells in a mouse, it'll cost me $1 million and I'll be put in jail for 10 years because I offended his sensibilities. He thought only God can do these things, and so he put a bill in the Senate for this, and I don't care which side you're on because this is not a partisan issue. But President Bush said, yes, the most egregious thing you could do is to make a human-animal chimera, that is, cells of both. But when I answered Sam Brownback's question in the Judiciary Committee of the Senate, I said, okay, which one of these brain diseases shouldn't we work as hard as we can, as fast as we can, to solve? Because I can tell you, every one of those diseases we can approach by taking brain stem cells from the diseased patient or make a mouse with the disease and test whether the human stem cells will work. So this is about something that all physicians take, the Hippocratic Oath. This is to say, I'm going to set my own religious and moral and political opinions aside because I only have one obligation, two obligations. First, do no harm, and I'll hit that in just a second. And second is make sure my patients and my future patients have the best possible therapy. Well, that's clear here. Okay, now I'm going to give you the bad ad. And if you can't read it from there, I'll just tell you. I'm this year president of the International Stem Cell Society, and in 2 weeks we're going to come out with a press release, a panel paper, and a website. Because if you just list stem cells and any disease you want on the internet, you'll come up with over 200 companies that are going to take your money and give you a phony stem cell therapy. So here's how you know whether it's likely to be phony. Watch out for anybody who says they have a kind of stem cell that can regenerate all tissues, because there's no such thing in adults. And no embryonic stem cell can do it yet. So that's the first thing. Don't go to the Dominican Republic to have a hole in your heart fixed with bone marrow they took from you in Naples, Florida, injected into your heart directly in the Dominican Republic, and then they let you go the next day. And also go without the $75,000 you paid for a therapy that can't cure you. And they're taking you away from, if you have a really end-stage disease, they're taking you away from your family and your friends, and they're taking the money away from you and your family and your children, and they got nothing for you. Now, it's hard to winnow away at them, but here's one thing. If they have one cell that treats everything, you know they're bullshitting, for sure. If they're charging you $50,000 to $75,000, that's another good indication. And if you say to them, show me the approval of a clinical trial that protected human subjects that led to this therapy, and they can't answer it, then you know they're just out for your money. Now, when I was a high school student working with Ernst Eichwald, there was a child down the corridor who had acute leukemia, and I thought that was the worst thing on earth. And we need to be able to treat those people. And that child and many others went searching for therapies that didn't exist then. And there were phony cancer doctors in Mexico, just like there are phony stem cell doctors all around the world. And they know how to escape detection, and they know how to escape malpractice. The patient I told you was a patient, hole in her heart, 21-year-old girl. Her heart stopped on the table, and they kept injecting. She gets out of the hospital. You know, if that happened here, you'd be in the intensive care unit and followed. They let her go home. She showed up at Stanford Hospital the next day, and I called the administrator when I was told about the case. They said, you know, we'd like to do something, but the medical treatment was in the Dominican Republic, even though the doctor is from the States, and that's outside our malpractice laws. So everybody wants to do something, and we'll try to globalize this. Now, this is going to be close to the last thing I say, maybe or maybe not. Cancers. So now remember, the definition of a stem cell is simple. It's the cell in a tissue or organ that by self-renewal can make copies of itself for life. And no other cell in the blood-forming system can make another copy of itself when it divides. Only the stem cells. Well, cancers don't come from Mars, they come from our own tissues. Leukemia is a disease where a cell pretends it's a stem cell. It can self-renew and expand its numbers, but now unregulated by the body, so there's going to be more. But still, it tries to make blood because it hasn't switched over from being a normal blood-forming stem cell that gives rise to a cell that makes, say, granulocytes, one kind of cell, and then become a cell that stops making granulocytes. It's just gone part of the way toward being fully cancerous or malignant. So, Tanisha Rea, who was my student, Sean Morrison, my former student, Mike Clark, a guy I recruited from Michigan, and I, in 2001 proposed this hypothesis that within a cancer you have what's called the tumor stem cells. And they'll self-renew to make more, but they'll still make daughter cells. Maybe there's only 10 tumor stem cells for every 90 daughter cells. So 10% of the tumor is dangerous, 90% are the daughter cells of the dangerous cells. All of the drugs we have now for cancer were tested for their ability to shrink a mouse tumor or some cells in a tissue culture dish by 90%. If you didn't pass that 90%, you didn't get to the next step of treatment. And then they're tested for toxicity. Don't kill the person. And then they go on the market if they're safe. And you have to have high doses of drugs that have bad side effects and so on. Well, we found that for both radiation and chemotherapy, the stem cells are relatively resistant, but their daughter cells aren't. So the cells that got shrunk were the cells that didn't matter. So it wasn't until we could purify a tumor or a leukemia stem cell that we could ask, okay, what's specific about that cell? What genes does it turn on? What genes does it turn off? What's interesting on its surface and elsewhere? So, about 2 years ago, studying mouse leukemias, using the same mutations that humans had, but we put them into mice, so it's the same old story, we found that we could put in a combination of two genes that over a period of weeks finally led to acute myelogenous leukemia, a disease that usually affects people increasingly as they age. A disease that's very hard to treat. Cure rate is very low. But by looking at these mouse leukemia stem cells, we found they had a molecule on their surface that told the rest of the body, don't eat me. So this is amazing. It was actually put in, we think, so that a young cell, like a young red blood cell, wouldn't be eaten until it was old. And since a red cell can't make new proteins, the copies of this protein are put on its surface. As they decay, finally allow the don't eat me signal to be gone. And they got eaten. They got eaten by big eating cells called big macrophage eating. This is what happens all the time. You take a perfectly simple concept, you turn it into Latin, so only the doctors can talk to the doctors, right? My fishing friend? That's exactly what happens. But we could call them big eating cells, but because I have to talk about this to scientific audiences, it's a macrophage. They're all over our body. And now we know a leukemia in mice has it. So we quickly tested human leukemias. Every kind of human acute myelogenous leukemia overexpresses the don't eat me signal. So if they have the don't eat me signal and they're sitting in a plate full of macrophages, they don't get eaten. But if they don't have the don't eat me signal, where are they? they're eaten inside of the macrophages, which digest them and kill them. It's that simple. So we said, well, if it's that simple, can we make a therapy from it? And we found out the leukemia cell has the don't eat me signal, and the macrophage that would eat it has a molecule that receives the signal that stops everything. But it's not important to stop everything unless it got started. So there's an eat me signal also. And the cancers that win this competition, this Darwinian competition, are the ones that start to express the don't eat me signal, which counteracts the eat me signal. Okay? So now let's find out if it's important. So we took authentic human acute myelogenous leukemia, isolated the leukemia stem cells pure, transplanted them into immune deficient mice, and they got leukemia. The human leukemia, in weeks to months. And then a medical student, Mark Chow, and my postdoctoral fellow then, Ravi Majeti, treated them with an antibody that doesn't block the don't eat me signal, or an antibody that blocks the don't eat me signal, so the macrophage now doesn't think there's a don't eat me signal, and they get eaten. So this clears them from the bloodstream. This is not a mouse leukemia. This is from a patient. And if you look at it after treatment, it clears the bone marrow. So this is leukemia, and this is after that treatment. So then we wanted to know, will it work for any other tumor? So there's a disease which at least some people in this room know about intimately, called non-Hodgkin's lymphoma. And there we didn't know if it had a don't eat me signal, but there was a drug out there, an antibody called rituximab, which, when you give it to a patient, leads almost always to a remission, but then it comes back. And the cells that come back are resistant to the antibody, the eat me signal. So we combined the antibody that blocks the don't eat me signal and the antibody that causes it to be eaten, and we cure with human primary lymphomas taken from the patient into the mice, where neither antibody alone cures. They delay the onset, like in patients, but if you put two at a time in, you get a synergy and it cures them. It's on an ovarian cancer. We isolated ovarian cancer stem cell. This is a radiographic way to look at it. If you put in the wrong antibody, it grows and kills the mice. If you put in the don't eat me, it blocks. Now this is a mouse antibody against the human cells that we're using. Works in breast cancer. So these are primary breast cancers, put in the mammary gland of an immune deficient mouse after anti-CD47 therapy. Clears it. So since that time, we found all of these cancers have the don't eat me signal. And most of the ones we've tested, at least in a plate with human macrophages,